Okay, we're going to start the chapter on bacteria, or chapter on Monera. This is what the syllabus tells us we need to know. I'll put this slide up at the end and we'll reflect back that we've covered everything. So to begin, so what do we know about microorganisms, bacteria and fungi? Okay, first thing is they're everywhere. We can't avoid them. They're in the air, they're in soil, in water, on your skin, inside you. Okay, they're everywhere. They're extremely small and we can only see them with a microscope. They're on an order of about 1,000 times smaller than a millimeter. They belong to a group of organisms called the Monera, the kingdom Monera. And we can see here the rules for the Monera is no nucleus. No nucleus or other membrane enclosed structures are allowed. So that's the general rule. No nucleus or membrane enclosed structures allowed in the kingdom Monera. They're also classified as being prokaryotes, which is another way of saying or cells without a nucleus. All of the other living things, plants and animals and fungi, they all have a nucleus. The general structure of bacteria, so their cells, and all bacteria cells have a cell membrane controlling what gets in and out of the cell. They have a cell wall for strength and support, and they have a cytoplasm where chemical reactions occur. Bacteria have ribosomes to make protein, and they'll have a large single chromosome. Human cells have 46 chromosomes enclosed inside a nucleus or with a nuclear membrane. Bacteria just have one large chromosome and it's loose inside the cytoplasm. So some bacteria have outer capsules which are there to protect the bacteria from stomach acid or from enzymes that a host organism might throw at it to destroy it. So the capsules give the bacteria protection. Many bacteria have plasmids, which are small loops of DNA with their own genes. These plasmids can actually be transferred from one bacteria to another. These bacteria in the picture have flagella, which are long protein strands that the bacteria can use to move. So under a microscope, it looks like the bacteria are swimming around as if they have a purpose. So these flagella are for movement. There's a picture from a Leaving Cert exam question. I've just added the labels. So be able to label a diagram like that. And if we can pause the video and draw that. Now here is the list of the structures on a bacteria. Okay, we'll need to pause the video again and jot these down. Okay, from the top, the cell wall provides support. It prevents the cell from expanding. Bacteria cells will tend to gain water by osmosis, and the internal turgor pressure could burst the cell if it wasn't for the cell wall. So they have a cell wall like plants, but unlike plants, their cell wall is high in nitrogen, and plant cell walls contain cellulose. The cell membrane is what controls the entry and exit of molecules in and out of the cell. The cytoplasm is where all the chemical reactions occur, and ribosomes make protein. Ribosomes assemble amino acids together in the correct sequence to make protein. The single chromosome is where the genes are located, and genes are sections of DNA, and they control the functions of the cell. Many bacteria have capsules on the outside. The capsule protects the bacteria from secretions of a host, from stomach acid or from digestive enzymes. The plasmids are small loops of DNA found in bacteria. They're incredibly important when it comes to studying genetic engineering. And the flagella, as we said, for movement. These are the different types of bacteria, so we can classify bacteria in different ways, but the easiest way to classify bacteria is by shape. So the three general shapes of bacteria. These are small circular or spherical shaped bacteria. They're called cocci. An example here would be the Streptococcus bacteria that causes sore throat. These are rod-shaped bacteria called bacilli. Example would be the bacteria in your large intestine. Okay, we can call them rod-shaped. or Bacillus. So if you're taking this down, you can put ROD, R-O-D, in brackets after Bacillus. And the spiral-shaped bacteria, an example would be a bacterial infection called Syphilis, which was a fatal bacteria infection, which is sexually transmitted disease that caused skin rashes and was fatal in the 15th and 1600s. Now we're going to look at how bacteria reproduce. The most important thing is they reproduce asexually. There's only one parent. There's no male or female bacteria. So just a single cell divides in two to produce two daughter cells. The daughter cells are usually identical to the parent. Okay, they're clones. But because there is a very fast rate of reproduction, variations within a population can build up. So in 24 hours, there could be as many as 70 generations of a bacteria. So variations can pop up. So this is how bacteria can evolve. And as we'll see, they can become resistant to antibiotics. The name for bacterial reproduction is binary fission. So binary fission is how bacteria divide asexually. So binary fission simply means splitting in two. If you're asked to explain the stages in binary fission, okay, we have these four stages. The DNA replicates, so the chromosome makes a copy of itself. The cell gets bigger, it stretches. Each copy of the DNA moves to opposite ends of the cell. And finally, the cell splits in two, so a separating wall forms and two new cells are formed. So that's the DNA replicates, the cell stretches, copy of the chromosome moves to each end, and the cell simply splits into two. Bacteria also have what I'd call a survival mode. If conditions become unfavorable, if it becomes too hot or too cold, or there's not enough food, or the pH is wrong, bacteria can go to a long-term survival mode. And this survival mode can last for millions of years. So these are when we form endospores. Endospores are structures bacteria use to protect themselves from unfavorable conditions. An endospore is basically a tough wall that forms around a chromosome. And the endospore just becomes dormant. And it'll stay dormant until conditions become favorable again. The oldest bacteria have been extracted from sand sediment that was dated to 250 million years old. And the bacteria started growing again as soon as it was extracted from the sediment. Now, we need to look at the stages that bacteria go through as they grow. So if a single bacteria lands on a food source, the initial growth rate will be quite slow. The numbers of bacteria are small. The bacteria is adapting to a new food source. So bacteria will initially go through what's called a lag phase or a slow growth phase. Once the bacteria have become established and they've adapted to the food source, that's when they'll start to grow very quickly. They enter then what's known as the log phase, sometimes called the exponential phase. So in this stage, the number of bacteria that are being formed is far greater than the number of bacteria that die. So we have a very, very rapid increase in population. The next stage is the stationary phase, which is the numbers of bacteria is stationary. The number of new bacteria equals the number that die. So something is limiting the population growth. It's being limited by space and food. Excretory wastes such as urea are building up, and that's reducing the number of new bacteria that are formed. This won't last. We'll eventually move into the decline phase, assuming space is limited and food is limited. The population will start to fall very quickly. So again, this is due to a lack of food and a buildup of excretory products, waste products. Not all bacteria will die. Some of them will survive. They'll form the endospores. So we'll have a small number of surviving bacteria. These endospores can grow again if conditions improve, if the pH improve, if food is becomes available again. Okay, here's a diagram you should take down. It shows the different stages: the lag phase, the slow growth at the start; the rapid growth is the log phase, graph rises up very quickly; stationary phase at the top, plateaus, no population change; and the decline phase on the right hand side. And off the screen, and maybe you might draw it in for yourself, is the survival stage, where some bacteria do survive, and you'll have a horizontal line again. Okay, you might put in time. I missed that. Put in time at the bottom. Okay, now I just want to show a short video on that.
It's all very well using powerful microscopes, but it doesn't really help you get a hold of just how small bacteria really are. A magnification factor of 1,000, 10,000, it doesn't really mean that much to me. But I've had an idea. An ordinary sewing needle. And this is a needle too, a very big needle. In fact, this is just the very tip of the needle. The rest is down there. It's 250 meters long. Now, if I scaled up a single bacterium as much as I have this needle 5,000 times, how big do you think it would be? This big? This big? This big? It's this big. Bacteria are just one group of microbes. Some of them are much bigger. Scaled up 5,000 times too, an algae would be this big. A single-celled organism like an amoeba would be this big, but some are much smaller. Even scaled up 5,000 times, a virus would still be smaller than a fine pinprick. But there's another way of seeing these tiny things. They may be too small to see individually, but you can see large numbers of them. To live, bacteria, like all microbes, need moisture and warmth, and to feed and respire. There are plenty of bacteria in my saliva. This agar is a good source of food. And this incubator provides just the right warm, moist environment for bacteria to thrive. When in such good conditions, bacteria don't just survive, they also reproduce very rapidly. Bacteria multiply by splitting in two. You start with just one, then it splits into two. Each of these in turn divides into two, and then two again. The numbers grow very rapidly. So just how quickly do they multiply? Imagine we started with one bacterium. After 20 minutes, it divides into two. After 40 minutes, there are 4 bacteria. After 5 hours, 20 minutes, 65,536. I thought 65,000 peas would have been a bigger pile than that. After 7 hours, over 2 million. And after 12 hours, well, just how many do you think there are? 69,000 million.
Okay, now we're looking at how bacteria get their food. We can divide nutrition in bacteria into 2 main ways. Autotrophic nutrition, this is where the bacteria make their own food. On the right hand side, heterotrophic nutrition, bacteria take in food by other organisms. If we look at the left hand side first, autotrophic bacteria. These are the bacteria that make their own food, and there are 2 main groups of bacteria that can do that. Chemosynthetic bacteria. These bacteria use energy from chemical reactions. They use that energy to make food. The example that you should know would be bacteria from the nitrogen cycle, in particular nitrifying bacteria. They obtain energy to make food from the conversion of nitrogen compounds, specifically when they're converting ammonia to nitrites. So we do the nitrogen cycle when we do the section on ecology. With bacteria in the nitrogen cycle, they can make their own food, but they use energy that they obtain from chemical reactions. And at the bottom left, photosynthetic bacteria. So these are plant-like bacteria, I suppose. They're not plants, but they obtain their food in the same way that plants do. They use sunlight energy to make food. The group that you should remember are called cyanobacteria. Sometimes they're called blue-green algae. Okay, these are photosynthesizing bacteria. On the right-hand side, we're looking at heterotrophic nutrition. These are bacteria that take in food from other organisms. I've seen this asked a number of times in exams where students are asked to name the two types of heterotrophic nutrition by bacteria. Saprophytes take in food from dead organic matter. These are the decomposing bacteria, the bacteria that will break down dead plants and animals. They're feeding on the dead remains of plants and animals. And parasites or parasitic bacteria take in food from a live host. And these will be the disease-causing bacteria. Pneumonia is an example. So any pathogenic bacteria that might live on or in a living organism is a parasite. Okay, if you want to pause that and take down that table, please. Now, what will make a bacteria grow, or what do bacteria need to grow? Okay, what are the limiting factors for bacteria? So bacteria growth is affected by temperature, by the amount of oxygen available, by the pH, by the external solute concentration and pressure. And we're putting this information in the table here. If we look at temperature, first of all, the enzymes inside bacteria are affected by temperature. Okay, so these enzymes work very slowly at low temperature, and they're broken down or denatured at high temperature. So bacteria would prefer to grow at optimal temperature. Unfortunately for us, human body temperature is an optimal temperature for bacteria growth, or the best temperature. Looking at oxygen concentration, now not all bacteria need oxygen. The bacteria that do are called aerobic bacteria. They use their oxygen for respiration. Anaerobic bacteria don't need oxygen. They're quite happy to respire anaerobically. And interestingly, obligate anaerobes, these are bacteria that can only respire in the absence of oxygen. They need to be in an area with no oxygen. And tetanus is the example of a disease caused by a bacteria that if it penetrates your deep muscle tissue where there's relatively little oxygen, it can grow quite well. We mentioned tetanus before when we spoke about the immune system, that tetanus is treated by giving injections of antibodies. So it's an example of passive immunity. pH affects the growth of bacteria. So a very high pH, such as bleach, or a very low pH, such as vinegar, will kill bacteria. External solute concentration. So outside the bacteria cell. A high solute concentration, such as salty water, will draw water out of bacteria cells by osmosis and kill them. So if you place a bacteria in a salty solution, the salt will draw water out of the bacteria. This is osmosis happening, and the bacteria cells die. And high pressure is another way of killing bacteria. So placing food in a pressure cooker, the high pressure damages the cell walls of bacteria and that kills them. So these are all methods that are commonly used to preserve food. So if we're looking at the top, preservation by oil and vinegar is often keeping out the oxygen. Storing in airtight containers is the same. Cold temperature, remember that bacterial enzymes work quite slowly in low temperatures. Heat treatment, bacterial enzymes are denatured or destroyed at high temperature. And adding salt and sugar to foods. This is your external solute concentration, is drawing water out of antibacterial cells and killing them. So in exams, make sure that you say you draw water out of the bacterial cells and don't say you draw water out of the food. Now, if we're looking at harmful bacteria, the syllabus specifically wants us to know examples of harmful bacteria. Okay, so we refer back to the idea of pathogens. Pathogens are microorganisms that cause disease. Typhoid and syphilis are two examples there. Typhoid is a bacteria infection. It can lead to fevers and vomiting. Syphilis is another bacteria infection. It can lead to skin rashes. They're both easily treated with antibiotics, but at one stage in human history, they were very serious and often fatal diseases. And bacteria cause food to decay. Now, looking at antibiotics. Antibiotics are medicines. These are chemicals. They're produced by microorganisms and they stop the growth of other microorganisms. It's a natural chemical that a bacteria or a fungus would produce in order to clear its own zone, to kill its competitors, so that it would have plenty of food for itself. So antibiotics were first isolated in 1928 by Alexander Fleming. He accidentally discovered the first antibiotic, penicillin. Okay. In the 30s and 40s, antibiotics were the wonder drug that prevented infection and cured a lot of illnesses. However, since 1987, there has been no real new discovery of antibiotics. All the antibiotics provided since then are just variations on the same antibiotic. So it's becoming very difficult to discover new antibiotics. And many of the antibiotics that we've had are no longer functioning. The reason is because bacteria are becoming resistant. So how does that happen? So during reproduction, the cloning, the binary fission of bacteria, it isn't always perfect. Sometimes we have mutations, and these mutations will lead to differences and variations in the bacteria population. So some bacteria in every generation will be different. And remember, every generation can happen every 20 minutes. So in 24 hours, you can have 60 to 70 generations. So in every generation, some bacteria become or can become resistant to antibiotics. And if the antibiotics are overused, it's these resistant strains that will survive, and they will become the dominant strain. These resistant strains can also pass on the genes for resistance to other bacteria by plasmids. So in a way, they can almost teach other bacteria that haven't mutated how to become resistant. Not finishing a course of antibiotics is one way that allows resistant strains to survive and regrow. Very often, the resistant strain would have been killed if the person completed the course of antibiotics. But often they feel better after 2 or 3 days and they stop taking the antibiotic. But if they continue to take the antibiotic for 5 days, the antibiotic would have killed even the mildly resistant strains. So the infections caused by resistant strains of bacteria, such as MRSA, are very hard to treat. And if these bacteria are found in hospitals, it can be very serious for patients that are already sick. So it has happened that hospitals have had to close to visitors because there's been an MRSA outbreak in the hospital. And these strains of bacteria are very difficult to treat. Doctors don't have antibiotics to kill them. Okay, we're looking at now examples of beneficial bacteria. Not all bacteria are bad or cause disease. Okay, some bacteria used to make yogurt. These are the lactobacillus. Lactic acid is needed to make silage. So when you collect grass and put it in a pit for the winter and cover it with plastic, you're making anaerobic conditions, you're removing the oxygen, and the type of bacteria that thrive there produce lactic acid as a byproduct. This lactic acid is a low pH and will preserve the grass. Lactic acid produced in the vagina is part of our general defense system. It will kill bacteria. And since 1978, we've been able to genetically modify bacteria to produce human insulin. So it's producing a medicine. The term bioprocessing means using bacteria generally to produce a range of foods. It could also include making medicines and other products such as artificial sweeteners and food flavoring. You can think of yogurt production as a good example of bioprocessing. Bioprocessing, two main types, and you need to know these two types for your exams. In batch processing, a fixed amount of nutrients are added to the bacteria in a vessel or container called a bioreactor at the start. The bacteria are allowed to go through all the stages, that should be 4 or 5 stages, and the product is extracted at the end. So the container needs to be cleaned out and reused, and could be used for a different purpose the following week. So to recap, in batch processing, a measured amount of nutrients is added to a measured amount of bacteria at the start. They're allowed to grow and go through all the stages in the bacteria growth curve, and the product is extracted at the end. In continuous processing, which is less common, nutrients are constantly being added. The bacteria are staying at the log stage or the stationary phase, and products are constantly being removed. This is a less common process, it's an expensive process. It ties up the bioreactor to one function for long periods of time. So the two main types of bioprocessing, batch processing and continuous flow processing. Now, this is the last slide, so we're reflecting back on the syllabus contents. So if we look at it from the top, the distribution of bacteria and fungi in nature, we've discussed that bacteria are found everywhere, in your skin, in soil, in water, in the air. We've looked at the basic structure of bacteria. You've drawn a diagram. We know what the plasmid is. It's a small loop of DNA. We know the 3 types of bacteria shapes. We looked at how bacteria can reproduce with binary fission. And we looked at nutrition, autotrophic nutrition, heterotrophic nutrition, and there's 2 examples of each. Remember the factors affecting growth, the temperature, the availability of oxygen, the external solute concentration, the pH. We understand that pathogenic means a disease-causing microorganism, and we hear examples such as tetanus and syphilis and typhoid and pneumonia. They're all bacterial diseases. Antibiotics, these are chemicals produced by microorganisms to inhibit or kill other microorganisms. The abuse of antibiotics in medicine is referring to the overuse of antibiotics that has led to resistant strains of antibiotics becoming dominant. The economic importance of bacteria. Bacteria are needed to produce medicines such as insulin. Bacteria are used to produce yogurts. And in bioprocessing, bacteria produce many useful products such as food flavoring or flavor enhancers. Examples of any 2 beneficial and any 2 harmful bacteria, we revert to that. Beneficial bacteria would be insulin production and perhaps yogurt production. Harmful bacteria, any 2 bacterial diseases such as typhoid and cholera. Okay, that's the end of this presentation.